Hello, everyone. I think it's time, and I'm Min Soo from Seoul National University Hospital.
অ্যাটেন্ড করা হয় না, মনে থাকে না।
Okay. So today, I would like to discuss about the targeted cancer
না
treatment,
রাস্তা ফাঁকা না এরকম।
and it can be quite. I would like to
হয়তো আরো 5-10
share
মিনিট
some
বেশি ইয়ে থাকে,
basic
কনজেকশন থাকে,
knowledges.
একটু কম।
And I think, okay. So targeted therapy is a cancer treatment that uses drugs to target specific genes and proteins. And I think Dr. Pupri, could you, if it might, think if you can mute. Okay, and drugs to target specific genes and proteins that are involved in the growth and survival of cancer cells. So targeted therapy can affect the tissue environments that helps a cancer grow. And so it is a part of a precision medicine and for cancer treatment, and the precision medicine has been increasing steadily as a key factor for the targeted therapeutic market. And as precision medicine takes into account individual patient genetic makeup, it helps in overcoming various limitations associated with the traditional cancer therapy. So today I would like to share some basic concepts of these targeted therapies. And first, we need to understand the cancer microenvironment. And it contains many different type of cells. It contains cancer cells and many different type of and many different type of white blood cells and fibroblast fat cells like adipocyte and other cell types. And it also has a blood vessel, which are made up of endothelial cells and pericytes. And lymph vessels also can be present at the microenvironment. And all of these proteins are embedded in the protein scaffold called the extracellular matrix, as you will know. And cancer cells have the ability to exploit their microenvironment and in many instances manipulate it in their way. And as a vice versa, endothelial cells and white blood cells can also, like these cells in the microenvironment, can also contribute and interact to have the tumor growth. But this can be, if you think in reverse, if you can inhibit the function of one of these or block the interaction between them, then you can actually suppress the growth of the tumor. That meaning that each component of this cancer microenvironment can be a target for cancer treatment. So what can be a potential targets and what are the targets in this targeted treatment? So briefly, target in the cell surface, protein known as a growth factor receptors are there, and these are found on almost every cell in the human body. And cancer cells specifically often have an overproduction of these growth factor receptor, or these growth factor receptors on the surface of cancer cells can be like faulty or overactive. So this can be the first target. The next can be a kinase. So after growth factor receptor triggers the activity of other protein, it can actually activate the protein inside, and these are called the kinases, and it does a cell signaling. And it is these kinases are an enzyme and are a group of more than like 500 enzymes that cause, it makes one particular chemical reaction, which is the phosphorylation. And this phosphorylation is, as you will know, is like an on and off switch for the protein. So it can activate or deactivate the protein. As a result, so these kinases can control the cell signaling, and the faulty activation of such kinases can induce cancer. Next target can be the CD antigens in the cell surface. And these are proteins found on the surface of white blood cells and on the surface of cancer cells that like causes leukemia, lymphoma, and multiple myeloma. So in these type of cancers, like CD antigen targeting agent can be this can be a one target for the treatment. And as upper mentioned, that tumors need a blood supply, hence they produce signaling proteins that trigger the growth of blood vessels by a process called angiogenesis. And this tumor angiogenesis is the name given to the process through which small vessels sprout new side branches that form a new blood vessels. So it enables the blood to reach the tumor cells and fuel its growth. So in most cases, the tumors have elevated status of angiogenesis because they have more demand of energy supply for the growth. And these process and signaling proteins can be blocked by a group of treatments like angiogenesis inhibitors. And there are other like parts, like some cancers are driven by hormones, such as like estrogen for breast cancer and testosterone for prostate cancer. So blocking these hormones, like using the hormone therapy, can be one for the target. And some cancers like contain these fusion proteins, like which you will know is the BCR-ABL. They can be blocked with kinase inhibitor. And our cells use this proteasomes to recycle old and unwanted proteins. So some cancers can be dependent on proteasomes. So it can be also treated with the proteasome inhibitors. And next is and in some cancers, there is a faulty in the DNA repairing process. And usually there is a miss in the PARP. So it can be like PARP inhibitors can be used to target this DNA repair process. And also the many, like there are PD-L1 or PD-1 like immune targeting immune targets. So like cancer cells have proteins on their surface like PD-L1 and it can suppress the white blood cells. And and cancer cells also can persuade other cells in their surrounding to produce the same protein, and they can avoid by the immune system. So like targeting immunotherapies can overcome this suppression and can be a target for the cancer treatment. Okay, and lastly, there is we can also use a cell target and attach to it like chemotherapy, radiotherapy, or toxins. So like drug conjugate can be used for the targeted therapy. And this is what we do, like radiotherapy, radioligand therapies are what we are doing. So today I would like to focus on some facts like for the cell signaling part and also the angiogenesis and like PARP inhibitors in the nucleus and also the immune side of the immunotherapies. So before moving on, we need to again know about then what kind of formulations are used for this targeted cancer treatment. And by their formulation, most targeted treatment agents currently fall into two categories, which are the cell surface protein targeting the monoclonal antibodies, or there are like kinase inhibitors. And like there are other non-kinase and non-antibody treatment, which are like hormones for breast cancer and prostate cancer, and PARP inhibitors and immunotherapies are like CAR T cell therapy, vaccines, and viruses are non-kinase or non-monoclonal antibody treatment. So I would like to introduce some more about this monoclonal antibody and kinase inhibitors. Okay, before you can see the naming of the targeted therapy. So you can, when you hear the name of the drug, you can actually know what kind of formulation it is made of and what are their targets and what are their source. So in case of like when the drug like Rituximab, which targets the HER2 receptor and used in the breast cancer, it ends with the MAB, which means a monoclonal antibody. And like in the gefitinib, like tyrosine kinase inhibitor, it is end up with IB, which means that it uses a small molecule with inhibitory property, which is a tyrosine kinase inhibitor. And like Trastuzumab, you can realize that it ends with Zumab, and which means that it is a humanized mouse made by. And also like, as you can see, there is a Trastuzumab, you can see there is a T inside the letter. So you can also think that it targets a tumor. And also like naming of the like tyrosine kinases, like gefitinib also is a tyrosine kinase, so it has a name of TI. And there are other like pembrolizumab, like or that kind of immunotherapies. As it targets the immune system, you can realize there is a LI or L in the name of it. So just when you see a drug, you can imagine what kind of target and source they are using. And so first, for the monoclonal antibody, it means that it has a exactly same clones, multiple clones of antibody. And this antibody is firstly, our body actually contains many B cells, and each of these have a copy of a unique B cell receptor on its surface. And these B cell receptors are used to recognize and connect with the protein and other like complex molecules on the surface of invading pathogens, such as like bacteria and viruses. And anything that the B cell receptor can connect with is an antigen. And when a B cell connect with the antigen, they are activated. And after it becomes a plasma cell, and it releases these millions the B cell receptors. And when these B cell receptors are released into the blood, they are now known to be the antibodies. And as you can see, antibodies are a large protein put together in a particular way. And it contains like 4 separate proteins, like 2 heavy chain proteins and 2 light chain proteins. And each B cell makes an antibody with a unique antigen binding region, and which is created from the ends of the heavy and the light chain. From this and variable region, antibody can have a specificity to the antigen. And many of the proteins so targeted by the monoclonal antibody treatments are growth factor receptors. These are like proteins that attach to growth factors and activate signaling proteins within the cell. And so this antibody can block the growth factor receptor by attaching to the growth factor receptors and preventing the growth factor from gaining access to the receptor. Or it can directly block the receptor's activity. And also what it can do, it can prevent receptors from pairing up. And this is, I will explain later in the growth factor receptor part, that pairing actually is an important part for the cell signaling. And it can attach directly to the growth factors and to prevent these growth factors to actually to bind to the receptors. And so another way is that antibody can kill cancer cells by attracting white blood cells and complement proteins. And when an antibody attached to a protein on a cancer cell, it can attract white blood cells such as like macrophage or NK cells. And macrophage can engulf and digest cancer cells through like phagocytosis, or it can actually destroy the cell by the lysis. And also, this antibodies attached to the cancer cell can also like attract the complement proteins, and it can attack it can attack as a membrane attack complex that can punch a hole in the cell membrane and kill the cell. And such a mechanisms are like the former mechanisms, like Rituximab uses that kind of mechanism of binding like A and B. And actually bevacizumab, the VEGF inhibitor, actually binds to the VEGF, the growth factor, and stops its binding to the growth factor receptor. And these the rituximab uses a mechanism of attracting white blood cells. Then you can have a question, is rituximab a immunotherapy? And it is could be quite confusing part. And as I mentioned, that this rituximab attracts white blood cells and to kill the cancer cell. So it uses the immune system. However, like many people for their nomenclature, when the drugs directly targets the immune system, then they call those like immunotherapy, like the immune checkpoint inhibitors. However, like here, like rituximab, although uses the immune system, in some cases, they are not called as a immunotherapy, but it can be confusing, so you can just understand their mechanism. So it attached to protein called the CD20, and it attracts and activates the white blood cells, and white blood cells kill the CD20 producing cancer cells. So, and it is maybe the first targeted treatment, and it is used widely for the lymphoma and like hematologic malignancy treatment. And one another way antibody can be used for the therapy is by drug conjugation. And like we are very familiar to that. And antibody fused to radioactive particles are used to treat hematological cancers. And these white blood cells are extremely sensitive to radiation and cancer cells of like leukemia and lymphoma. So this means that radio-labeled antibodies can be an ideal treatment for these hematologic malignancies. And radio-labeled antibodies can also be used to suppress the patient immune system prior to their receiving a stem cell transplant. Though this is only relevant for hematological cancer in which like stem cell transplants are used. And currently, two anti-CD20 radio antibody therapy, the yttrium, the Zevalin and Bexar, are approved in the USA in the early 2000s. And like from many like clinical studies, it has proven the safety and efficacy of both Bexar and Zevalin. And like compared to the Rituximab or like the paper as in the consolidation setting. However, Bexar actually exited the market in 2004 and because of the low sales, and Zevalin remains on the market, but our sales are very low. And so it is, although the efficacy and safety was proven, and we might need to consider not only the efficacy, but the practical issues like price and availability for the production of drugs. So right now, these two drugs, although they are efficable, are limitedly used. And there are also other targets for the monoclonal antibody, like the blood vessel endothelial cells, the VEGF, which is the growth factor, not the receptor, and immune cells like PD1 in the immune cell or PDL1 in the macrophage, or like tumor cells can be targeted by the monoclonal antibody. And the pros and cons of, so why do we use this monoclonal antibody is that they are actually very precise. And an antibody designed to attach to one antigen is always attached to that exact antigen. And also, like as we mentioned for the drug conjugation, it can be used as a transportation devices to deliver toxic drugs like radioactivity and chemotherapies. And these monoclonal antibodies can be broken into pieces and stitched back together to create entirely new proteins. And many millions of identical copies of antibody can be reliably manufactured. So it can be easily modified, or we can have a tailor-made function of antibody, and it can be easily made. And they are very stable and last in the body a long time. So they can be given to patients as like weekly or even monthly treatments. However, there are like some like cons, like antibodies used as our cancer treatments are foreign to the patient's body. So there can be allergic type reactions like fever, chills, or headache. And because these antibodies are large proteins, they cannot cross the cell membrane like other like small drugs. So their target must be on the cell surface, as you have seen. And it actually limits the range of proteins in the cancer cells that can be targeted with this monoclonal antibody. And because of the size, it is hard to penetrate through the body tissues and like blood-brain barriers. And since it is a protein, it can be digested by gut enzymes, and therefore they have to be given by intravenous infusion or subcutaneous injection to ensure their injection into the bloodstream. And although the antibodies are very precise, but sometimes the proteins that they target on the surface of cancer cells can be also on the healthy cells, like growth factor receptors are overproduced in the cancer cells, but they are also in the healthy cells, which are like producing like skin. There is also like growth factor receptors, so it can also have a side effect in those kind of healthy cells. And next, widely used the formulation of the targeted drug is the kinase inhibitors. And as aforementioned, kinases are enzyme and catalysts that approach postpate to other proteins and molecules in our cells. So kinases have a docking site for both ATP and one more like substrate. And in this case, this is a inactive protein. And when both ATP and this inactive protein dock with the kinase transfer one of their phosphate from ATP to a tyrosine or threonine or serine amino acid in the protein. And when this phosphate are transferred, this protein activated, and we call this process a phosphorylation. After the phosphorylation, it gets active, and this active proteins are released by the kinase, and it further transfer for the cell signaling. Okay. As you can see from the image, like growth factor receptors are large protein that sit in our cell's membrane, and as you can see, they have three parts, which is the outer extracellular domain, which has a receptor part, and growth factors can be attached to this receptor part. And there is a transmembrane domain, and there are also intracellular domain. And here, there is two important features like docking site for ATP and multiple amino acids. And when ATP slots into this docking site, the growth factor receptor phosphorylate each other on tyrosine. So it can phosphorylate this tyrosine amino acid in each other side. And when there is a phosphorylation and this growth factor receptor become active, and it can deliver a signal to the further signaling pathways. So as you can see, this growth factor receptor also is a kinase. So it also can be inhibit by the kinase inhibitor. So, and also because it is a cell membrane receptor, it can also be inhibited by the monoclonal antibodies. So the way the kinase inhibitor works is that usually the drugs mimics the shape of the ATP binding site. So it mimics the shape of the ATP, and it can bind to this ATP binding site and compete with the ATP to bind. However, there are not only this kind. There are 4 classes of kinase inhibitors and like drugs that block kinase indirectly. So the type 1 kinase inhibitor compete with this ATP for the ATP docking site of an active kinase. And type 2 kinase inhibitor can enter this same ATP binding site, and only when the kinase is inactive. And it also contains a extra pocket. So it compared to this type 1, this type 2 kinase inhibitor has somewhat higher selectivity. And another is this allostatic inhibitor can be bind not in the ATP binding site, but outside of the ATP binding site. So this actually can be more selective compared to like type 1. And for D, it uses a covalent inhibitor. So it uses a chemical bonds. And when it enters to the ATP binding site, it forms a chemical bonds and hold the drug in that place. So it can be very stable when they are targeted to the drugs. And there are also like indirect kinase inhibitors, and they do not actually target their kinase itself, but it can indirectly, they can attach to a separate protein that only inhibits the kinase when the drug is present. And some examples of these indirect are the everolimus or temsirolimus, and which are quite, for the everolimus, we are using it for the treatment of neuroendocrine, the pancreatic neuroendocrine cancers. Okay. However, there can be the resistance for these kind of kinase inhibitors, and the mechanism might be more complex. But in some case, if there is a mutation in the ATP binding site, you may imagine that these 3 kind of kinase inhibitors may not work in such a cancer cell which has a mutation in the ATP binding site, and it will have a drug resistant. So now you can think of some, so you have seen different types of these kinase inhibitors, and you can imagine what might be the most, what might have more toxicity than others. And you might imagine easily that type 2 and this allostatic inhibitor, which has a relatively high selectivity to the target, will have less toxicity to the healthy cells. Since, but however, like when you target the ATP binding site, unfortunately, many of our, not only the cancer cells, but normal cells also have a similar ATP binding site. Therefore, these kinase inhibitors can also block these healthy cells. So it can actually cause a severe and cause a side effect. And moreover, it can be more serious for the covalent inhibitor because it has a characteristics of more stable and binding, but since it is irreversible, it can have more severe side effects. Okay. So the targets of these kinase inhibitors can be like very diverse. And since it is not like that big monoclonal antibody, actually it can penetrate inside the cells, and it can target the intracellular proteins. And like, as you can see, there are many kinases in the cancer cells, and the kinases can be overactive because they are present at abnormally high levels, or because they are faulty because of their gene mutation. And for some examples, there can be a mutation or overproduction or overactivity of EGF, like growth factor receptor, which is also kinase, and JAK or PI3K or AKT can have a mutation. So these mutated tyrosine, these kinases can actually produce the signal to the cells and make the cells like overactive, like a cancer cells, and it can be targeted. And there are also like kinases that are overactive, not because of themselves, but because of some mutations from the upper part. Like LAS or LAF or like AKT mutation will give a overactive signals to this MEK or mTOR. So it can be overactivated, and also it can be a target for the treatment. And not only in the cancer cells, but it can be also found in the cancer microenvironment. There are many kinases, and mostly you can think of is in the angiogenesis part. And here for the angiogenesis, there is a VEGF, the receptor in the endothelial cell. And this VEGF receptor also, because it is a growth factor receptor, has a kinase domain. And so this can be blocked by multiple like kinase inhibitors. And some characteristics of kinase inhibitor targeting this VEGF is that it can target many other, not only VEGF, but other like growth factors or kinases. So usually it has a characteristics of a multi-targeting inhibition. And they also have like pros and cons, as you can think. Like kinase inhibitor, because of their size, can cross the cell membrane, and it can have a target in the intracellular domains. And as in the same vein, because they have a small size, they can more easily travel around the body than compared to the monoclonal antibodies. And it is a chemical compound, so it can be taken in a tablet form, and you can actually get this kinase inhibitors, not like IV injection, but like oral tablet can be manufactured. And as mentioned, because it can block, it have a multipotent, because ATP binding sites, there can be many similar ATP binding sites for the different kinases. So it can block more than one kinases, and it can have a unexpected additional use for this kinase inhibitor. However, this can be also a problem for the side effects. And some problems are that it is, compared to the monoclonal antibody, it is quite imprecise for the targeting. And because of mutations, it can have easily a drug resistance can happen. And since it is a chemical compound, it is broken down by the body very quickly. And it has a high degree of drug to drug and patient to patient variations. So, so when you get a kinase inhibitors, you have to get like this bi-daily or even twice daily for the treatments. Okay, so we have seen the two most widely used formulations of this targeted treatment, the monoclonal antibody and the kinase inhibitors. And now let's talk more about the targets. And the first part is about the cell signaling, and it happens in the cancer cells and also like blood endothelial cells. And usually in the cancer cell or the blood endothelial cell in the cancer microenvironment, these cell signalings are like overactive or overproduced or have a faulty effect, making the cancer overgrowth. So blocking the cell signaling can be an effective way for the treatment. So how does this cell signaling work is that first, growth factors bind to the growth factor receptors, and it is phosphorylated and it is activated. And after the activation of growth factor receptor, it controls a number of different signaling pathways, such as the MAPK, the mitogen activated protein kinase pathway, and the PI3K, like mTOR pathway, and JAK-STAT pathway are like three main signaling pathways. These many proteins in this signaling transferring proteins are called as an intermediary proteins. And from using these pathways, the signal is passed to the proteins at the end called the transcription factors in the cell nucleus. And these transcription factors then attach to the genes and cause a gene transcription and protein production. So, like in short, when growth factors attached to the receptor, this ultimately cause the cells to alter it can control the survival, proliferation, and movement of the cell. And when this signaling process goes wrong, cells can overgrow and turn into cancer. However, in the same time, so it can be a target for the cancer treatment. And so the first target can be the growth factor, which is in the cell membrane. And when how it works is when the growth factors attach to their receptors, the receptor change their shape and pair up with each other. And once these receptors are paired up, they phosphorylate each other. And then it gets activated and it can attach to their partner on like these like phosphorylation of two receptors change their shape and docking sites for the proteins in the intracellular domain. So here the proteins can bind to and give a signal to the inner part. However, so the drug, when you target a growth factor receptor, there can be a two targets, as I mentioned earlier. There is a binding site in the extracellular domain and also a binding site for intracellular domain. And so like for the receptors, you can use a monoclonal antibodies, and for the intracellular domain, you can use a kinase inhibitors. So these are many examples, some examples of the drugs targeting the growth factor receptors like EGFR and HER2, and also like in the blood cell, the endothelial cells like VEGF or other growth factors like RT can be also targeted. So quite commonly used the agents are EGFR inhibitors, which is like Rituximab, and also EGFR kinase inhibitors can be used. Mainly these are quite widely used for the treatment of non-small cell lung cancers, like gefitinib and like Tarceva or other drugs can be used. And also there are HER2 inhibitors, which can be used for the treatment of like breast cancer, and also in HER2 mutated stomach cancer patients, like trastuzumab, and there can be a trastuzumab emtansine, which is a drug conjugation with a chemotherapy. And it can also, like, there can be a kinase inhibitors. However, when targeting the growth factor receptor drugs, it can also induce a drug resistance. And this resistance mechanism is by, firstly, what I have explained, there can be a mutation and altered ATP binding site. And in most cases, there is a mutation in the T790M their gene mutation. And then like ATP binding tyrosine kinase inhibitors cannot like match to it. And also there is because there are additional growth factor other than the EGFR. So there are like HER2 or MET or FGFR. So these alternative pathways can be used. And also it contains cancer cells can contain a like overactivated B-Raf or PI3Ks, or loss of like NF1 or P10, and meaning that so the inner signal pathway can be overactivated by itself, regardless of the growth factor receptor status. So this can be in this kind of conditions, the cancer cells have gain a resistance to the growth factor receptor targeting drugs. So there can be some way to overcome this resistance. Firstly, you can make up like now there are third generation drugs which can target this mutated EGFR tyrosine. So these inhibitors can target the mutated part. And also you can use a drug conjugated EGFR, like targeting like trastuzumab emtansine, which conjugates a chemotherapy in the trastuzumab. So when it binds with the receptor, not only it stops the EGFR, but it can give the drug inside inner cellular portion and have a killing effect. And next is the inner signaling pathways, the MAPK signaling pathway. So it is conduced of LAS protein and LAP and MEK or ERK proteins. So it are like intermediary signal transferring proteins. And so you can, when there is a faulty or overactivity, you can actually block these. However, currently, there is no drug targeting the RAS protein directly. So like if you can make one, we think that RAS protein targeting agent might be very effective, but there is currently no drug. Alternatively, there are drugs that can target like RAF or MEK. And you will be familiar with the drugs, the B-Raf inhibitors, because these B-Raf inhibitors are quite widely used in the thyroid cancer patients. And as you can see, that the popular thyroid cancer patient contain quite a lot of patients contain a B-Raf mutation, like in here up to like 40%. So in case of these patients, like B-Raf inhibitors can be used as a targeted inhibitor. And also similarly to the EGFR, this B-Raf inhibitor can also gain a drug resistance. So by like, if there are many other like alternative pathway problem, like PI3K, like faulty overactivity of the NRAS or like other, like not then BRAF, but CRAF or like PI3K, it can have a resistance. And also like overproduction of like CRAF or BRAF can have a resistance. And like here, the cancer cells can actually modify the BRAF. And so like making a shortened, the truncated version of the BRAF. So then this inhibitor cannot be attached to it. So these are some mechanisms of how the, like for the B-Raf inhibitor, how the cancer cell can have a resistance. And the next part is of the PI3K and like mTOR signal pathways. And the paired up receptors, as you will know, activate the PI3K, and it can indirectly activate AKT. And this AKT has a numerous impact on cells, like including like it can trigger cell proliferation, survival, and it can alter the metabolism. And also it affects the mTOR, and mTOR also causes further activation of AKT itself. In addition, so mTOR also has, it can have a response to the cell's well-being, like environment, such as changing levels of glucose, amino acid, and ATP, and oxygens. So you can think that when you target such a signaling pathway, there is a change in the cell's metabolism directly. So like glucose metabolism can be changed when you treat a patient with this in the targeting the signaling pathways. So there has been some attempt that to show that FDG PET can be used for the early treatment response for prediction for this targeted treatments. And in this paper, they have shown that when so these cell lines are gefitinib sensitive cell line, and when gefitinib was treated, you can see that there was a decrease in the cell glucose metabolism, and like hexokinase activity was also decreased. So you can see for this, the gefitinib sensitive cell line tumor model, you can see that right after the treatment, there is a significant decrease in the glucose metabolism. So they proposed that it can be used for the early response prediction. And so, like, there has been papers that it can be used for lung cancer patient, for like tyrosine kinase in EGFR inhibitor, the prediction of their efficacy. So like in the patient 3, you can see there was a early decrease in the uptake, the glucose metabolism. And you can see that this patient, although SD, had a quite long PFS. However, like for patient number 5, you can see that there was a early increase in the glucose metabolism. And as you can imagine, this patient also had a PD in the follow-up. So, and also there are like other papers regarding the comparing the FDG PET and CT to evaluate the response after the EGFR inhibitor. And it proposed that, as you can see, that this FDG PET used metabolic response was better to predict the overall so actually this was to predict the pathologic response. So patient underwent neoadjuvant therapy, and after the surgical resection, they have seen the pathologic response. And here they found that FDG PET can better predict the pathologic response after this targeted therapy. And also there are papers regarding that in the RCC patient undergoing the tyrosine kinase inhibitor, this FDG PET can be used for the prediction. Okay, so this was part for the cell signaling, and we might have some break, and then we can move on to the angiogenesis and other part. Okay, so I think we can have 10-minute break. So let's meet again 10 minutes later. And in between the lecture, if you have any questions or comments, please let me know, and you can just put on a chat or just ask me directly. So there are, I'd like to again introduce some mechanism of how angiogenesis inhibitors work. So for example, bevacizumab is a humanized monoclonal antibody, and it attached to the VEGF-A, and it keeps away from this VEGF-A to attach to the VEGF receptor. And there are also like a bioengineered protein that attach to the these growth factors. And also there can be a humanized antibody that directly attached to the VEGF2 receptor. And there can be also, as we have mentioned, a kinase inhibitors that can block the kinase portion of the VEGF receptors. And so these are some examples. And as you can see that some small kinase inhibitors that targets the VEGF receptor have not only the VEGF for the targeting, but also they have multiple receptor targets. So these kinases that targets the VEGF inhibitors can have a characteristics of multi-target inhibitors. And you might be familiar with like lenvatinib or sorafenib because these can be used for the thyroid cancer, like and other drugs can also be. And however, there can also be these drugs can also have a resistance, and there are some mechanisms which all could be quite different from the other like EGFR resistance mechanism. So actually, the angiogenesis inhibitor can cause oxygen levels to drop. And when the oxygen levels drop, it can trigger tumoral cells and endocellular cells to produce even greater amount of like VEGF and PDGF or like angiopoietins, like growth factor. And so these growth factors can activate the endocellular cell receptors, keeping them like alive and triggering yet like more angiogenesis. And also there can be some cancer cells contain DNA mutations and other adaptations that enable them to survive low oxygen levels. So in this mechanism, these cancer cells can be insensitive to the angiogenesis inhibitors. Okay, next is about the fusion protein. So before, you might need to know about what is the fusion proteins. So each of our 46 chromosomes in our cells contain like hundreds of genes, and cancer cells often contain a broken chromosome. And sometimes these two chromosomes have broken at the location of two different genes. And as the cell repair the damage, it might accidentally put the chromosome together the wrong way. And when the cell transcribes this fusion gene, it can create a strand of mRNA that contains information of this wrongly fused gene. And this ribosome can, using the mRNA, make a fusion protein. And since this is from a wrongly made fusion genes, this fusion protein can have a faulty action. And for example, the translocation or other chromosome rearrangement involving the ALK gene forces the cell to make a uncontrollable like ALK fusion protein. And a mutation affecting the kinase part of the ALK has made it overactive. And this implication of this overactivity forces the cell to make more ALK genes. And so the overactivity of the ALK triggers a numerous signaling pathways which force the cell to grow, multiply, and stay alive. So these are how the wrongly made fusion proteins work in the cancer cells. And usually these fusion proteins which induce such a cancer has a kinase part. So then it can be inhibited by the kinase inhibitor. And the well-known targets and fusion and well-known fusion proteins which contains the kinase are the BCR-ABL or ARK or ROS1 or RET. And this also can be a target for the thyroid cancer. Okay, so next I would like to introduce some briefly about the targeted therapies used in the thyroid cancer. And the thyroid cell growth and the proliferation of it are regulated by the multiple pathways, signaling pathways such as the LAS MAPK and PI3K and JAK-STAT pathways. The activity of this receptor tyrosine kinase in the thyroid cancers are dysregulated. And so this dysregulated tyrosine kinases can be a therapeutic target for tyrosine kinase inhibitors. And the TKI range in selectivity from broad spectrum multi-kinase inhibitors to very specific inhibitors. So for like multi kinase inhibitors, you will be well known of like lenvatinib, sorafenib, or vandetanib can be a multi kinase inhibitor. And there can be more selective, like which has a direct B-Raf specific inhibitor. And also there can be a RET specific inhibitor, like these drugs. So what you need to check is that such a multi-kinase inhibitors can be used in the thyroid cancer patient regardless of their mutation status, because they can target like many growth factors and VEGF. However, these more selective tyrosine kinase inhibitors should be used according to the gene mutation status of the patient. So there can be, this is a brief how you do the treatment for the metastatic differentiated thyroid cancer refractory to the standard treatment. So in this case, what you do if the patient is refractory to the standard treatment, you then need to see whether there is a targetable driver mutation. And firstly, if it is no, what you can do is you can use a this multi-kinase inhibitors. And what people suggest is for the first line of this multi-kinase inhibitor is widely used is the lenvatinib for the first line. And it has proven it as you can see, the median progression-free survival was 18.3 months. And compared to the lenvatinib, which is 3.6 months. And there was a progression-free survival benefit in all pre-specified subgroups. And the response rate was rating up to 65% lenvatinib group. And there was like also complete response was seen in these patients. And there are also other options instead of lenvatinib. Like there can be you can use a sorafenib, which targets VEGFR and platelet-derived growth factor receptors and RAD PTC subtypes and B-Raf, and also vandetanib inhibitor that targets can be used. However, as you can see, compared to the lenvatinib, the sorafenib and vandetanib has somewhat quite lower, but still it lower like the median survival, progression-free survival. However, still, it can be used for patients. And however, like this Vandatinib is available in the United States for the treatment of unresectable metastatic medullary thyroid carcinoma. And it is actually not approved for the popular PTC patient. However, it can be considered as a clinical trial, or it can be a treatment option to consider for a patient with a radioiodine refractive progressive PTC patient. So the most widely used right now are firstly, the Lenvatinib will be the first one. And then you can also use Sorafenib can be used as a multi-kinase inhibitor in the patient where the mutations are not identified. However, if there is a identified targetable drivable mutation, you can use such targeted inhibitors. Like if there is a NTRK fusion mutation, you can use these kind of drugs. And if there is a RAT PTC oncogenes, you can also use a RAT directed inhibitors. And also, like there are, since this B-Raf kinase inhibitors can be used in patients with B-Raf V600 mutant PTC patient. So yeah, these can be a option for patients who have a identified mutations. Okay. And the next part is about the PARP, the poly-ADP ribose polymerase inhibitor. And actually, these PARP inhibitors were originally made for the people, the breast cancer patient who have a inherited a mutation in one of their BRCA genes. So you need to understand why this PARP inhibitors was made for BRCA mutation patient. And before that, you might need to know what the PARP are doing. So PARP, it has a function to repair a single strand breakage in the DNA. So if PARP is well working, it can directly repair, or in some case, if there is a double strand breakage, then this BRCA protein works to repair this double strand breakage. So, and this process is called a homologous recombination. However, in some case, if a woman is born with a fault in one of her BRCA gene, then because of other second healthy copy of gene, she is keep healthy, but there can be a damage of one copy of gene, then the second copy of gene, then this one, it can make a it can make it errorless repair of the DNA, and because it has a very error-prone repair mechanism, so it actually this kind of wrongly repaired gene and proteins can be accumulated and can give a sign to overgrowth of the cell, which in turn can make a breast cancer, or it can make a ovarian cancer. Then how can this PARP inhibitor can have a effect in this BRCA gene mutated breast cancer is that if the woman is treated, like breast cancer is, women is treated with a PARP inhibitor, this prevents our cells from using PARP to repair single-strand breaks. And these breaks ultimately become a double-strand breaks, which the cancer cells can only repair using a error-prone method. And so the cell quickly accumulates the DNA damage to the critical levels and the cell dies. So, however, the rest of the healthy cells in our body, which still retain a healthy copy of BRCA gene, can perform the double-strand breakage repair, so can be stay alive. So it means, so because of like very sudden increase of the DNA damage when you do the PARP inhibitor, it can kill the cancer cells, but it can still make a healthy cell alive. So it can have like very targeted effect in such BRCA gene mutated breast cancer patients. However, another thing you can think about this PARP inhibitor is that because it acts as a single-strand breakage repair, so you can think of like that these, like our radiation also has an effect by single-strand breakage or double-strand breakage. And if they are not repaired, or if the repair is faulty, this radiation effect can be like more encouraged. So you can think of combining this PARP inhibitor with a radiation therapy. And actually, there was a study they use a in vitro 3D models of neuroendocrine tumor cells, and this use with the PARP inhibitor, they have shown that it can the tumor suppression with the lutetium dotatate induced a very critical, and was shown that it can be a promising approach for improving the therapeutic effect, as you can see here. So there are now ongoing trials for the combination of lutetium dotatate with the olaparib, which is a PARP inhibitor, in the treatment of GAP-NET patients. Okay, the last part will be the immunotherapy. And as I explained, these therapies are targeting directly the immune system and altering the system. And the mechanism is that directly the backward of how the cancer cells escape. So firstly, cancer cells can be eliminated by the immune cells. However, when they grow and grow, it can have a mechanism to escape, like by low levels of MH class 1 and antigen pros, and like it gets low levels of tumor cell recognition in this stage. And then it escapes the immune action. So the immunotherapy actually goes to reboot the immune system to cure or kill these cancer cells. And there are like different mechanisms that are used. And the first can be the checkpoint inhibitors. And the majority of checkpoint inhibitors are monoclonal antibodies that attach to the checkpoint proteins or their ligands. So it can disrupt the interaction between the checkpoint protein and its ligand, and it can increase the activity of the white blood cells. And there can be another is a adaptive like T cell transfer. So you can purify the T cell from patient's blood and then modify it in the laboratory and then infuse back into the patient where they can attack the cancer cells. And there can be also like peptide and DNA vaccine can be if you inject a DNA or a peptide into the skin or muscle, it can be picked up by an antigen presenting cell. And this antigen presenting cell displays the peptide on its surface. So making it to activate the T cells and our immune system against this tumor-derived antigen. And there, lastly, you can have a bispecific antibodies these are modified antibodies that create a physical link between the cancer cells and T cells, like triggering the cells killing activity of the T cell. So these are some strategy and mechanism how immunotherapies are now ongoing. And the most widely known immunotherapy for now is the immune checkpoint inhibitor. So it works like the activity of this cytotoxic T cell is suppressed when there is a inhibitory checkpoint proteins. And inhibitory checkpoint proteins can be CTLA-4, and it can be inhibited by the ipilimumab, or there are others like in the PD-1 and PD-L1, and these are more in the tumor microenvironment. So there are like PD-1 directed inhibitor, which is nivolumab and pembrolizumab, or there can be a PD-L1 inhibitor like avelumab and atezolizumab. So the goal of checkpoint inhibitor is to overcome or remove things that the suppressing part, like PD-1, PD-L1, and CTLA-4, to and increase the number and activity of the cancer-killing T cells naturally generated by the patient's immune system. However, you can imagine that it uses the patient's immune system. So if the patient's immune system is not well functioning, this immune checkpoint inhibitor may not work that much sufficiently. So when you need to check the immune status when giving this kind of treatment to the patient. And it has proven their safety and efficacy from many clinical trials. And as you can see, there are now like 7 drugs that were approved by the FDA for like 2021. And there are more in Europe and Asia. Another problem is how to choose the patient to give this immune checkpoint inhibitor. And what might be the predictive marker was there, and people are using, because it is targeting the PD-1 and PD-L1, the first predictive biomarker will be the PD-L1. However, there are problems of the pathologic biomarkers, which is reliability of the detection method, and there is still no, there should be a consensus of reasonable cutoff level, and there is a interlesional heterogeneity. So however, what you get is a one site of a tissue. And by the treatment and because of the immune status, the PD-L1 expression status changed dynamically. So this pathologic based PD1 or PD-L1 expression provides information of one part of the tumor at one moment. So there had a emergence of the need for imaging biomarker, and so a group had made a zirconium labeled atezolizumab, and it has published a article. Oh, I'm sorry, this is wrong. The references are wrongly indicated. So it has been published in the Nature Medicine. And so they have shown that using this zirconium atezolizumab, it can well predict the patient response. And it had better prediction compared to this two type of IHC, PD-L1 IHC compounds. So it had better predictive level. So there are many ongoing clinical trials to test this immuno-PET imaging to see the predictive value or whether it can be used as a biomarker for the immunotherapies. So in a recent, we might get this kind of immuno-PET in our clinical practice. And there are other treatments such as tumor infiltrating lymphocytes or like CAR-modified T-cell therapies. So, and one thing is that this tumor infiltrating lymphocyte is actually using is based on the patient's immune system. And so it can be quite similar to the concept of the immune checkpoint inhibitor. However, this CAR-modified T cell is quite different from the concept of the checkpoint inhibitor. The goal of this CAR-modified T cell is to create genetically modified T cells that can kill the cancer cell. And this genetically modified T cell has a characteristic that it doesn't exist in the patient's body. So it is it is regardless of the patient immune status. And it can attach to one particular protein on the surface of cancer cells. And when it attaches to the receptor, it can activate and kill the cancer cells. So, so people are thinking it can be a very good treatment option for the patient. So, like as you can see here, a modified virus containing the gene for CAR protein is used to genetically modified T cell taken from the patient's blood. And when there is a CAR, this modified T cells can be amplified and then given back to the patient to have a therapeutic effect. Okay, so this might be the end of the lecture. And so today we have summarized about the targeted treatments now used in the treatment of cancer. And most of the targeted treatments fall into two categories, as you will know, the monoclonal antibody or kinase inhibitor. But there are others like hormones or CAR T-cell therapy or PARP inhibitor are non-antibody, non-kinase inhibitor. And cell signaling pathways go awry in cancer cells, and they can be target. So they can be also the target for cancer treatment. And firstly, we have seen how the growth factor receptors can be a target, and other inner intracellular part signaling pathways like MAPK, PI3K, or JAK-STAT pathways can be target for the treatment. And for the growth factor receptors, it's also in the endocellular cells of the blood vessels. So VEGF also can be a target, and it can also be a target to inhibit the angiogenesis. And and also fusion proteins, because it contains kinase domains. It can be also inhibited by the kinase inhibitors. And PARP inhibitor PARP is used to repair single-strand breakage in the DNA. And this PARP inhibitor can be effective in patients with BRCA gene mutation, such as breast cancer and ovarian cancer patients. And also, like Offer mentioned, that PARP inhibitor can have a emerging effect with the radiation, and for us, the radioligand therapy. And lastly, the immunotherapy is emerging, and there are lots of drugs and indication for this immunotherapy, and the market is keep growing. And so we also think that expect that immunoPET can be a helpful biomarker for predicting and selecting the patient who are going on for the immunotherapy. And my slide were mostly referred from this book, The Targeted Cancer Treatment. So if you would like to get further information regarding or more about to understand more about targeted cancer treatment, I think this book can be very useful. Okay, thank you very much. And would there be some questions or comments?
Hi, Professor Su.
Oh, hello.
Thank you for your interesting presentation. I have one question. I'm very interesting about the PARP inhibitor that inhibits the DNA repair. As we know that lutetium PSMA in prostate cancer only cannot disrupt the DNA completely. So can we combine the PARP inhibitor after lutetium PSMA? So maybe the survival will be longer.
Oh, yeah, I think so. There can be we can design a trial to, actually, I only have just to look up for the, like any dotatate, but there might be a clinical trial, ongoing clinical trial for the PSMA also, but I haven't looked up in the, so I think I should. And there are also like concept about not only the PARP inhibitor, but about the BRCA mutation. So like as we have thought that BRCA protein acts as a double-strand breakage repairing. So if there is a mutation in the BRCA, there will be a faulty repair for the double-strand breakage. So and prostate cancer patients have quite a lot of BRCA mutations. So there are also like some conceptual works that whether this BRCA mutation status may have an impact on the PSMA therapy. So that is also, and I think the PARP inhibitor will have a emerging like synergistic effect with the PSMA mutation therapy also. Thank you for the question.
Thank you very much, Professor.
Okay, I think if there is no further question, I think I can we can stop and we can meet next week. Okay, thank you very much for all for the participation. And
thank you very much.
Yeah, thank you very much.
Thank you very much, Sir Hamida.
Thank you.